Test, test. Hello. Good afternoon, everyone. Thank you for your patience. So we are about to start a study union session. So for those of you in the back of the ballroom, you probably want to move up if you're here for this session. If not, we're going to have to ask you to leave for now because we do need the ballroom for this session. Okay? And then we are here for microbiology, general microbiology. It was supposed to be held by Jessica, but unfortunately she's not feeling well. So just so you know, you are in the correct place if you're here for general microbiology, okay? Stephanie's just following. I'm sorry, covering for her. Any questions? Perfect. We're about to get started. Okay, thanks. What time is it? What is it? I don't know where that is. Okay. We'll just keep an eye on time. Hey everyone, my name is Stephanie, so I'm going to hold this review for you guys because Jessica's out. But she left us the final review for her that she created. There's 21 questions. It's located at the SI Google site. Where was it? Never mind. Sorry, not this one. The SI Google site under Biomedical Sciences, and then if you go down to Microbiology Jessica, you'll find Microbiology Final Review Doc. So this is where you can find your own, like the sheet, so that you don't have to copy down questions or anything like that. So this review is 21 questions long. We're going to go through all the questions, all the answers, and get those answers for you. And then we're going to end around 10-15 minutes early so that you guys can ask your own questions that maybe we didn't get to go over. But feel free to raise your hand with questions related to the number that we're on. Yeah. The answers don't appear to be on the SI site. It looks like the final review, and then that's about it. So we'll go over answers today in this review. Okay. And then maybe later they'll be posted again, I'm not sure. But this session is recorded and live streamed, so if you want to see this again and you want to go over the answers in case they're not posted again, you can go to the YouTube page for UCF SARC, and it'll be posted there by today. Okay. So we can get started. Number 1 says, in a human, how does the number of body cells compare to the number of bacterial cells that compose the normal flora? Okay, so now we're comparing the cells in our body compared to the bacterial cell that's usually in our body. So we look at our choices. So our choices have A, there are 10 times as many human cells as there are bacterial cells. B is just the opposite. There are 10 times as many bacterial cells as there are human cells. And then C and D are roughly the same, but now instead of 10 times, we're looking at 20 times the difference. And then E is there is no correlation that can be determined. So based on your notes, we can know that bacterial cells are more abundant than human cells. So we can cross out C and A. Let's say human cells are more than bacterial. And then from your notes, I think she has on her PowerPoint slides that there are 10 times as many bacterial cells than there are humans. So this one is more of a you remember what's on the notes rather than figuring it out and trying to piece together the information. So this is more of a simple memorization type question. So I would check your notes on this though, because I know that like more recent research kind of doesn't show this correlation as well. So that's why I was also looking at it and thinking E could also be right. But you want to go based on Dr. White's notes rather than outside information. So I think according to her notes, it should be B, that there are 10 times as many bacterial cells as there are human cells in the body.
number 2 says, clownfish live in anemones, which provide them protection from predators, while the anemones are unaffected by the habitation. This is an example of A, mutualism, B, parasitism, C, commensalism, D, A, and C, or E, none of these. So in order to answer this question, we need to first kind of define mutualism, parasitism, and commensalism. So mutualism sounds like mutual. So a mutual agreement is where people both agree together and things are good. So mutualism is kind of where both the clownfish and anemone would benefit. It'd be a mutual type interaction. They'd both be happy. But in the question, it says that the clownfish are being protected. They have that positive benefit, but the anemones don't have any positive benefit. They don't have any negative, but they just don't have any positive. So that's why mutualism probably wouldn't be the best choice for this one. And if we take out A, that means we also kind of take out D, because D is A and C. Okay. B says parasitism. So a parasite would be something that's going to harm you, something that's going to harm the host. So like leeches or parasites, they suck your blood out, or mosquitoes, things like that. So parasitism would cause a negative effect on the host. So the clownfish, although it's having a benefit, the anemone isn't having a negative effect. It's just unaffected. It's neutral. So because the anemone is neutral, it's not positively affected or negatively affected. A and B are both not good. Not the best choice. C says commensalism. Commensalism, I kind of look at it and think like, oh, it's common, or they, I don't know, like you can kind of either remember the definition or kind of walk your way through what it sounds like it would be. And this one's kind of where one benefits but the other just isn't hurt. So this would be our best choice, because the clownfish is benefiting but the anemone isn't being hurt or benefited. So it's not mutual, it's not a parasite, it's commensalism. The anemone is just unaffected. So number 2 should be C. Number 3 says, which of the following is an example of an opportunistic pathogen? So an opportunistic pathogen is going to probably not hurt the host right away, but when they have a chance, when kind of the guard is taken down, they're able to then attack and kind of be seen as a pathogen in that case. So that's kind of opportunistic pathogen. They find the opportunity and then they seize it. So these could be your normal flora. So under normal circumstances, they don't really do anything. They're not harmful, but if you kind of kill away other ones, like so if you get an infection and you take some antibiotics and you kill away, you try to go after the bad ones, taking away the bad ones and some of your good ones may allow for some of your normal flora to kind of go in and take over. So for opportunistic pathogens, we're looking for something that wouldn't normally be a problem but becomes a problem. So if we look at our answer choices, A says, a person becomes sick due to salmonella poisoning from undercooked chicken. So this isn't necessarily an opportunistic pathogen because you're eating the pathogen and then you're getting sick from it. So it's just right away infecting you. It's not under certain circumstances that it's able to infect you, but normally it wouldn't. So salmonella usually under any circumstance would have an effect due to food poisoning. That wouldn't necessarily be an opportunistic pathogen. B says a person gets the flu after an outbreak at work. So this one's similar to A, where the flu is a virus, so you're going to acquire that from someone else, from touching dirty things and touching your eyes or your mouth or any other area to get infected. So this is more of a straightaway, like, you get infected and then you're affected by it. It's not just like certain circumstances that allow this pathogen to take over. Because usually if you have a flu virus, you're going to show symptoms unless your immune system or immune to it. C says a person gets a yeast infection after a treatment with antibiotics. So this one's a little bit different. So this one, the person was being treated with antibiotics, and then because that's kind of taking away bacteria, that's kind of destroying the competition, this yeast infection is allowed to occur. And this would be an example of an opportunistic pathogen, where you take away the competition and then it's able to cause problems. So number 3 would be C, for an opportunistic pathogen. Do you have questions so far? Are we okay? You good? Okay. 4 says, all the following are enzymes involved in the evasiveness of pathogens except. So these are a list of our enzymes, and we kind of have to know what these do and if they're involved in invasiveness. So we have to know what invasiveness is as well. So invasiveness is, kind of sounds like invade, so it's how these bacteria or pathogens are able to break down barriers and invade a host. So these help them do that. They help either break down connective tissue, break down things that hold cells together, and things like that. So A is hyaluronidase. Can't really say it very well. But this enzyme, if you remember, is the one that's going to break down the cement, they call it, between the interstitial space between cells. So like the area between cells, they're breaking down that connective tissue and being able to allow that pathogen to invade the host. So this is, for invasiveness, this is a spreading factor. So hyaluronidase is considered one, so that's not one of the excepts. This is one that is true. B is lipase. So lipase kind of sounds like lipid a little bit. It's the enzyme that breaks down lipids. And if you remember, some membranes, usually made of lipids, have a lot of, like a high lipid content, as well as proteins and other things as well. So this one also is involved with invasiveness, breaking down. So it's like lipase is usually an enzyme that's being secreted, and then it breaks things down, allows the pathogen to invade. So both A and B so far are for invasiveness. Let's keep going. C is collagenase. So this breaks down collagen. Collagen is a connective tissue in the body. It helps create muscles and bones and other tissues. So this is breaking down that protein, breaking down the tissue, and allowing them to invade as well. So this is also another one that's involved in invasiveness. And then we have fibrinolysin. So fibrinolysin is breaking down the fibrin, so fibrinolysin. So lysin is lysing it. Actually, lyse is usually breaking down a cell. But the fibrin, so fibrin is usually found in blood clots. So when you get a cut and you're bleeding, you usually clot and form a scab to kind of keep things out. So this enzyme or protein is allowed to break down that fibrin, break down the blood clots, and kind of get in. So the answer for 4 is actually going to be all of these, because they're all used in invasiveness. So breaking down physical barriers to attack a host. Invasive. Okay. 5 says, if one species of bacteria, A, has an LD50 of 50, and another species, B, has an LD50 of 100, what conclusions can be drawn? And this one is choose all, apply, or check all that apply. So that's always fun. LD50 stands for lethal dose. 50. So lethal dose 50 means the amount that will cause the death or kill 50% of a population that they're testing. So usually they test it on mice and things like that, and they see like what's the minimum amount they can give this population to kill off 50% of them. So we're looking at, so A only requires 50 of those cells. B requires 100 of those cells. So B requires more cells to kill the same amount of animals or organisms. So in keeping that in mind, we'll look through the answers. So A says species A will kill half of a test group with 50 cells. Species A has an LD50 of 50. So that one sounds good because it will kill half a test group, and LD50 is 50% or half. It's using 50 cells, and the LD50 is 50, that means 50 cells. So A is good so far. B says species B will kill half of a test group with 100 cells. With species B, it has an LD50 of 100. So LD50 is still lethal dose to kill 50% of a test group or a population. So LD50 is 100. It says species B needs 100 for killing half a test group. So B is good as well so far. C and D, so one says A is more virulent, and D says B is more virulent. So either one of these can be correct, or none of them, because either one's more virulent than the other, or they're the same. Those are kind of our options for this. Since they have different LD50s, they're going to have different virulence. They're going to have different magnitude of their effect. So the one that's going to be more virulent is going to be the one that takes less cells to kill the same amount of animals or same amount of a test group. So for species A, it only requires 50 cells, and species B requires twice that much. So because species A only requires 50, it's more virulent than B because it doesn't require as many cells to do the same amount of damage. Okay. So for choose all apply, our answers should be A, B, and C. Species B isn't more virulent because it requires more cells to do the same amount of damage as species A. Okay. 6 says, all the following are true of endotoxins except. So we're looking for that exception. We're only given answers, so we don't have like none of the above, all the above to look at. So we're trying to find which one would not be true. So first, we kind of want some more information on what an endotoxin is, where we find it. Endotoxins are from gram-negative bacteria. We're going to find that in the lipid A portion of the lipopolysaccharide layer, or LPS layer. So this is only in gram-negative. It's part of the extra layer it has over its cell wall, the LPS layer. So in order to get at the lipid A layer, we kind of have to break down the cell to do that. It's not just going to be able to release these endotoxins by itself. It's part of its extracellular layer, and it's not just going to be able to give off those lipid A layers without breaking itself down. So endotoxins are that. We can also kind of compare them to exotoxins, which I think it does in the answer choices. So let's look at the answer choices. A says endotoxins are released during cell lysis. That would make sense because cell lysis is when we're breaking down the cell, and these endotoxins are found in the LPS layer, and we kind of have to break up that LPS layer in order to get to them, in order to have them kind of spread and wreak their havoc. So it's kind of like a defense mechanism almost. So endotoxins are released during cell lysis. That one would be true. That one sounds very good so far. As one of those that are true, because endotoxins are contained in the layer. We have to get to them. B says endotoxins stimulate fever in hosts. So this is kind of you remembering all of the symptoms of endotoxins, and if you're not sure, we can always skip this one and go back. So that's always an option on tests. But endotoxins do stimulate fever and other inflammation, great amount of inflammation in the body. So that one's also technically true. C says endotoxins are primarily found in gram-negative bacteria, and that is also true because endotoxins are part of the lipid A portion of LPS. Gram-positive bacteria don't have an LPS layer, so they don't have a lipid A portion of an LPS layer, so they don't have endotoxin. So that is also definitely true. So then we can look at D. D says endotoxins are more toxic than exotoxins. And just from knowing that A, B, and C are correct, we can kind of pick that one. But also, or just remembering, I think there's a chart in your notes that compares endotoxin effects and exotoxin effects. And it is true that exotoxins are going to be more lethal and more toxic than your endotoxins. So D is not good, not the right, or not, is false about endotoxins. And since this question is asking which of the following are true except, this will be our exception. So A, B, and C are good, D is not, yeah. I think with more toxic, they're thinking about like more lethal and what will like kill the host really quick. So although like endotoxins are slow to produce effects and they have really drastic effects, they're not usually as lethal as exotoxins, which are much quicker and a little bit worse. Other questions? Are you okay? So 6 is D. 7 says, which of the following cells are involved in adaptive immunity? So immunity that has to be adapted from your environment. That's not innate immunity. So it's kind of the opposite. Innate is what naturally occurs. Adaptive would be you need to go through processes to actually create this immunity. So adaptive is also known as like specialized immunity or specific versus non-specific, that kind of thing. So which of the following cells are involved in adaptive immunity? So I have choices A, lymphocytes, B, neutrophils, C, phagocytes, and D, macrophages. So with this one, B, C, and D are kind of similar because C is phagocytes, which is really general. Macrophages are a form of phagocytes, and so are neutrophils. They both do that, so phagocytes are going to engulf the foreign bacteria and break it down. And those two are involved in that similar thing. So phagocytosis of foreign cells or foreign bacteria, foreign particles, that antigen is going to be part of that non-specific type immunity where it just sees something bad and it kind of gets rid of it right away. Versus in lymphocytes, so lymphocytes you have your T lymphocytes and your B lymphocytes. In your T lymphocytes you have cytotoxic T cells, helper 1 and helper 2 T cells, and then those B cells are going to form those antibody-producing plasma cells. So your lymphocytes are where you're going to find your antibody-producing cells. And antibodies are very specific to a certain antigen, a certain pathogen that's entered the body. So when we're looking at what's involved in adaptive immunity or specific immunity, our answer choice is going to be A, because that's where we're going to find that antibody production, that specialized immunity. The other ones are just kind of finding things that are wrong and just kind of engulfing them, and that is part of the innate, that non-specialized type of immunity, or it doesn't require certain knowledge of what that bacteria is, of how to handle it. Then we have number 8. So number 8 says, which of the following is an example of adaptive immunity? So again, this is the kind that requires those antibody productions and things like that. So looking at what's going to cause the body to produce antibodies. So if we look at our answer choices, A says a person becomes infected with the chickenpox virus and fights it off. B says a person immunized with the mumps virus vaccine becomes exposed to an outbreak. C says a mother transfers antibodies to her baby through breast milk. And then D says B and C, and then E is all of them. So when I first look at this, I look at C and I say a mother transfers antibodies to her babies through breast milk. That's a transfer of antibodies. So like the baby has those antibodies from the mother, but the baby's not producing them. The baby's not going through the adaptive immunity process and finding the new antigen and creating antibodies because of it. It's just borrowing the antibodies from its mom until those cells and those antibodies are eventually going to be taken out and kind of die off. So it's very short-lived, this type of immunity. So this is more transfer. So this is not a type of adaptive immunity because it's not stimulating the immune system to produce its own antibodies. It's using someone else's. This is a form of passive immunity, where you're just passively accepting others' antibodies and then using it as your own. So sometimes that also includes like blood transfusions for people who cannot make their own antibodies and need someone else's to fight off infection. But that doesn't last very long because they can't replenish them. So C is not good, and that kind of cancels out D and E, because if C isn't good, then B and C can't be good, and all of them can't be good. So then we're kind of left with A and B. So A says a person becomes infected and then fights it off. B says a person is immunized and then becomes exposed to the same thing again. So it's like the second time around, it's meeting that virus. So the second time around, when you're infected with something that you've already built up immunity to, your body isn't going to react exactly the same way. It doesn't have to go through the whole process again of trying to figure out how to create antibodies for this bacteria or virus, because now it knows the virus a little bit. It has some of those memory cells. So if you remember, those B cells, those B lymphocytes, are not only going to be activated and produce, become plasma cells that produce antibodies. Some of those plasma cells that produce antibodies are then going to become memory cells, so that when they see that virus again, they're able to quickly activate and attack and be able to produce antibodies for it. So when we compare someone that's seeing a virus for the first time versus someone that already has that immunity built up, when we're looking for what's adaptive immunity, that one's going to be the one that's first introduced to the virus. So if B was different, B is saying that a person is immunized and it just stops there for the virus and doesn't become exposed again. So when it first becomes immunized, that's when the person's undergoing adaptive immunity and creating antibodies to fight either the weakened virus or the inactive virus, whatever the vaccine is for. So because they're seeing that antigen again, or that virus again, that's why this wouldn't be a good example of adaptive immunity, because they're seeing it for the second time versus the first time where they're making antibodies. But in A, a person is newly infected with chickenpox and is fighting it off. That means they created those antibodies to fight it off. They've gone through adaptive immunity. So 8 is going to be A for this one. says, all the following are characteristics of innate immunity except. So innate immunity is the immunity that you have without going through the process of creating antibodies. This is the non-specific immunity. That's the first kind of defense against other, against pathogens. So innate is, I guess, innate in this case. So it's always there, and it's the first defense. And it's non-specific, as opposed to adaptive immunity or specific immunity. So if we're talking about innate immunity, let's go through the answer choices. A says it's specific to general groups of microbes. B says it's the first line of defense. C says it's very diverse. And D says it lacks memory of previous infections. And we have the question is, all of them are true except. So when we think about innate immunity, it is the first line of defense. So B is good. A says it's specific to general groups of microbes. So this is the non-specific immunity type. So it's not necessarily specific for a microbe or specific for a type of virus or bacteria. So that's why A doesn't look like a good choice, but we'll look at the others. C, it's very diverse. So I'm thinking, I mean, they have a bunch of different ways to attack an innate immunity. And they do have different cells. They have different macrophages to attack, or not macrophages, but phagocytes. They have macrophages, neutrophils, and other phagocytic bacteria to attack. And also, like, not just phagocytes, they also have, for innate immunity, your barriers for your skin and your mucous membranes and other things like that are also part of innate immunity. So it's not just cells that are part, there's also the physical barriers and chemical barriers, like enzymes you have in your tears, lysozymes, to help break down bacteria as well. So this is all part of innate immunity. So this is usually, yeah, so I think that's what they're talking about when they say very diverse, is that you have cells, you have physical barriers, you have chemical barriers, and that's kind of what they're talking about with diverse, and that is true. So first line defense, they're very diverse. They have cells, chemicals, physical barriers, and then D says they lack memory of previous infections, which is also true. We don't make memory cells, because if we made memory cells for previous infections, then that would be a characteristic, something that's specific, because now we're like, oh, we remember that one. This is what we use for that. And this is more like a one solution fits all type thing when we talk about innate immunity. So for innate immunity, B, C, and D are true. First line of defense, very diverse, and they lack memory. And then A is going to be the exception. They don't have specific groups of microbes for that. Yeah. Number 7. They are in that case, but they also are involved in innate immunity. So they're kind of a cross between the two, because the T helper cells are going to be presented the antigens from the phagocytes and then kind of induce cytokines and other kind of messengers to tell them to continue attacking those type of antigens. So they're kind of both, but they are also nonspecific. Like they will start attacking without presenting antigens to lymphocytes. Versus lymphocytes, the T cells and B cells require an antigen to start activating and creating antibodies. Those things won't happen automatically. Phagocytes are involved. Technically, yes. They're right, but they're not the best answer. So this one's more about like which one is the best answer compared to the others versus which one's right and which one's wrong. So these phagocytes and macrophages are another form of phagocyte, and those usually are antigen presenting. I think there might be a list, not a list, it's kind of broken up in your notes of what's antigen presenting and what's not. But I think most phagocytes are antigen presenting, so those will present antigens to the lymphocytes, and those would then use that to help make a specific response. So, but in order to get those antigens, they have to attack whatever's invading the host and break that up and do that on their own. So that's kind of part of the innate, where they take it upon themselves to just start cleaning up and battling whatever's infecting, and then they kind of go over to the adaptive immunity section and kind of get them going. So they're kind of a bridge between the two. But for this one, the one that's most correct for adaptive immunity would be lymphocytes, because that involves the B cells, and that involves antibody production, which is very much adaptive and non-specific, and specific, not non-specific. But yeah, that was a good question. That was definitely like a gray area, but this is definitely only adaptive and not innate. Other questions for 7, or adaptive versus innate immunity? We okay? Okay, so 7 was A, and then 8 was only A, and then 9 we were on. And 9 we said that the exception to this is going to be A, because it says specific to general groups of microbes, and innate immunity is not specific. It's non-specific. They're both like same term, or different terms for the same thing. Okay. So then we have 10. It says, which of the following antibodies are involved in the production of an allergic reaction? And our answer choices are IgG, IgA, IgD, IgE, IgM. So this is kind of remembering what our functions are for our different antibodies or different immunoglobulins. So immunoglobulin or Ig and antibody are the same thing. IgG is the one that's usually most abundant in your system. Yeah, it's most abundant, especially during that secondary response to a pathogen. So like in question 8, when it says a person was immunized and then exposed again, IgG would be in the highest concentration in that instance, because it's that second time around that it's seeing that same thing. So that's what usually IgG is for, as well as some other things. IgM, I believe, is that first time around, that initial infection. So like in 8, when it says a person is first infected with chickenpox and fights it off, that first response, or that primary response, is going to be, I think I remember from IgM. So IgM is part of primary response, IgG is part of secondary response, and it's usually the most abundant in your system. IgA is the one that's usually for secretions in your body, so like in your tears or in your saliva and things like that. So not for allergic reaction. IgG, I don't know if she really goes over that much about function for IgD. I think it's mainly the structure of it. But for this one, IgE is the one that's usually for allergic reactions. I think that one is also for parasites, if I'm not mistaken. That one's going to, involved in that inflammation and fever type reaction from allergic reaction. So for this one, you need to kind of know the functions of your immunoglobulins, and which ones are going to create that allergic reaction, and that one should be IgE. Yeah, so for the immunoglobulins, I usually make a chart on, so I can compare them all. So you compare also not just the function of them, but also their structure, because sometimes she may ask questions about their structure, like which one contains a J chain, how many units do they have. So like IgG would be just one divalent type immunoglobulin, but the others are usually more than one. So some have 5, some have more than that, and you kind of want to know how many units it has and things of that nature as well. So like kind of good study habit to kind of create charts and compare and contrast your different immunoglobulins so that you don't get them confused and mixed up. But for 10, it was D. 11 says, which of the following are true of Th1 cells? So Th1 are T lymphocytes, here T helper 1. So T helper 1 are going to be involved with the activation of cytotoxic T cells or TC cells. Yeah, so T helper 1 is usually involved in TC cells, or cytotoxic T cells. They help with that. And they also are presented antigens by phagocytes, such as macrophages. T helper 1 is, or T helper 2 is the T lymphocyte that is the helper 2, and that one helps with the B lymphocytes or B cells and activates them to create plasma cells that are producing antibodies. So T helper 1 is specifically for cytotoxic T cells and activating those. They don't activate them directly. They both kind of are presented antigens by a phagocytic cell, and then when a cytotoxic T cell is presented an antigen on MHC complex 1, and the Th1 cell is presented an antigen on MHC complex 2, Th1 is going to release cytokines that are going to help activate the cytotoxic T cells. Because cytotoxic T cells need the antigen presented. It's a little squeaky. Okay. Anyway, we'll ignore it. Maybe. Okay. Sorry. Cytotoxic T cells need two things to be I know. So I'm like, I don't think I will be able to shout and make him stop. But he pauses every now and then, unless someone wants to like tap him on the shoulder. Your chair is really squeaky when you rock it. Sorry. But thank you if you would kindly not do that. What? Oh yeah, I'm going to repeat it because I'm not. Oh, repeating number 10 or like what I'm talking about right now? 11, yeah, okay. So we didn't fully go over it because I was trying to like compare Th1 and Th2 because I don't know if she goes over Th2 in here. Yeah, she doesn't really talk about Th2. So T helper 1 involved with cytotoxic T cells and activating them. Cytotoxic T cells need two things to be activated. They need an antigen presented with MHC class 1, which is a protein complex. And then they also need a cytokine to be released from the Th1 cell. So they need two things to be activated, and then they can go attack sick cells and get rid of them through apoptosis or cell-mediated or cell death, programmed cell death. So Th1 is involved with cytotoxic T cells. Th2 is going to help, or Th2 is going to help with activating B cells. So B cells are phagocytic. They're going to present antigens on MHC class 2, and the Th2 cells are going to then activate the B cell to produce plasma cells, or to become a plasma cell. The plasma cell is what creates antibodies. So the unactivated B cell doesn't create antibodies. The plasma cell creates antibodies. In order for a B cell to become activated and become a plasma cell, it needs to present an antigen to Th2 cells. It presents the antigen on MHC class 2. So T lymphocytes can only read antigens from those with MHC class 2. So those with MHC class 2 are going to be phagocytic cells like macrophages and B cells. But cytotoxic T cells are going to be the ones that interact with MHC class 1. And MHC class 1 are on all cells in your body except for like the non-nucleated ones. So like mature red blood cells won't have a nucleus in them. So they won't have MHC class 1 anymore. So Th1 and Th2, so helper T cells are going to interact with MHC class 2. Cytotoxic T cells interact with MHC class 1. B lymphocytes are going to have MHC class 2 because they are also phagocytic. That's how we compare Th2, Th1, and B cells. So this question is just on Th1 cells, so they help activate cytotoxic T cells, and they also interact with MHC class 2. So A says they stimulate production of cytokines that suppress macrophages. B says they stimulate production of cytokine IL-2 or interleukin 2. C says both are true, and D says neither are true. So for this one, we have to know exactly what's right and what's wrong, or else we're going to get this one wrong. So A says they stimulate production of cytokines that suppress macrophages. Suppressing would be limiting them and not allowing them to be active in immunity, and that would not be good for the host or the cell or the body. So they're not going to produce cytokines that are suppressing. They're going to produce cytokines that help macrophages become more active. So they're not suppressing macrophages. They're doing the opposite, because the macrophages are going to help phagocytize the invasion or the invaders and the pathogens. So A is not good. So far, if A isn't true, then C is also not true because it says both are true. And so far, A is not good because we're not suppressing macrophages. We want to help actually get them moving and do the opposite. B says they stimulate production of cytokine IL-2 or interleukin 2. So this is kind of where you have to remember which interleukin is produced from T helper 1 versus the interleukins that are produced from T helper 2. In this case, T helper 1 does produce cytokine interleukin 2. So B is true in this one. So if you make a chart just like comparing all of your T lymphocytes, like cytotoxic T and T helper 1, T helper 2, one good thing to note would be what kind of cytokines they release. So for T helper 1, it's releasing interleukin 2. T helper 2 is releasing, I think, like 2 or 3 others. I think IL-6 and some others. I'm not sure off the top of my head, though. So you can look them in your notes. But 11 is going to be B, just B. They stimulate production of interleukin 2. Oh! Yes. Yes. I did not read that correctly because it says all following true except. Yes, yes. See, she got me. She got me. But she didn't get you. You got that one. Okay. So the A is false and B is true. But this is an except question, so it has to be the false one. So actually it's A. Thank you for catching that. I wasn't going to catch that. Okay, so we look for the false one because it says except. It said it in capital letters, so I should have seen that. But it's okay. So that just tells you to read the question completely instead of stopping and just assuming what it's asking you. Sometimes I have a problem with that. But you guys, your eyes aren't going to fall for that on your test because now you're very aware that it could just trip you out. So 11, not B. It's A because A is wrong, and we're looking for the exception, and A is the exception. Because we're not suppressing macrophages, we're doing the opposite. But good catch, good catch. Then we have 12. 12 says all the following are potential vaccine sources, except. So A says an attenuated strains of viruses. Sorry, vaccines, we're looking at viruses. B says active strains of viruses. C is pathogenic products. D is viral protein coats. And E is all of them. So this is all the following are potential vaccines except. So this is what is not. So we're looking for the one that's wrong, unless all of them are wrong. I think that's what it's saying. If it's except and we say all of the above, does that mean all of them are exceptions? I don't know. That's kind of weird. Anyway, so look at our answer choices. We have attenuated strains of viruses. So we need to know what attenuated means. From my memory, that is weakened. Yes? Yes, yes? Okay. So weakened strain of a virus. B is active strain of a virus, which would be very dangerous because they didn't do anything to a virus. They're just like, here, you can have an infection. That doesn't sound good. So attenuated sounds okay because that's kind of what some vaccines are. They're weakened strains, so you're able to kind of produce antibodies and overcome that. But if you kind of give someone the active strain the full force, that's not necessarily going to always work out 100% of the time. Get away. Then we have pathogenic products. So things that that virus is going to make or that pathogen produces. So that would also kind of be probably not a bad choice because maybe they can't attack, maybe like the pathogen itself isn't that bad, but what it produces, it's like the harmful part. So that's not necessarily false. D says a viral protein coat, which, so viruses are usually that protein coat and then the RNA or DNA that's held inside. So if we just use the protein coat, then the body can make antibodies that can identify the outside of the virus versus just what the inside does. And when you are first introduced to the virus, your body is going to recognize structures on the surface of the virus or on the protein coat. So this would actually be a good way to make a vaccine, to just kind of give the viral coats to a person or give a weakened strain to the person, or even those pathogenic products, because it's not the full-on bacteria. We're not going to, or virus. We're not creating the products continuously. We're just having a little bit of the products, and then you can kind of overcome those on your own. Or we have a weakened strain of the virus, so your body is able to overcome that. Or you just have the protein coat, so your body can attack that, and it's not really going to attack back and make other viruses. It doesn't have the genetic information inside. So all of these look like good potential vaccine sources, except for this active strain of the virus. This active strain of the virus is that full force virus, and that's what we're trying to make a vaccine for. We're trying to help people get immunity against that. And if you kind of give them the active strain, it's just like they're getting infected any other way. That's why they say to get a flu shot versus go get the flu and deal with it that way. So if the flu shot was just an active strain of the virus, people wouldn't be advertising the flu shot so much versus just getting the flu as it comes. So when it says all the following are potential vaccines, except, the exception would be the active strain. So you don't want to just infect people with the active strain of a virus in full-fledged force. So attenuated is fine because that's weakened. The body can overcome that. Just the protein coat is fine because it can't replicate other viruses and cause a full-blown infection. And then the products of a virus would be fine as well. So the body can attack those, and they're not going to be like multiplied, like if you just had the actual virus continuously generating those products. So 12, the exception is going to be B, the active strain. Then we have 13. 13 says all the following are examples of sterilization techniques except. So we have to kind of remember all of these techniques and know if they're sterilization or not. So this is more of a, do you remember from your notes or can you figure out here? So autoclaving is usually how tools are sterilized in the lab. They're put in an autoclave. So this is a sterilization technique. Pasteurizing is a way to sterilize fluids and bacteria by using heat. So pasteurization is another technique. Then we have UV light and ionizing radiation. And I believe from your notes you'll also see UV light and ionizing radiation as forms of sterilizing technique. Yeah. Okay, so is it just Yeah, so that'd be one of those gray areas, because it does get rid of some, but I guess not all in sterilization. Yeah. Okay. So then if we have, like, the book says that, I think you can go with that. I would also check her notes to double check, because sometimes they're not completely the same, and I think she tests more on her notes than the book. So I would double check with her notes. But if the book explicitly says that pasteurization, although you're killing off most bacteria, you're not killing off all of them, and sterilization is to kill off all of them, then B would be the exception for that, yeah. Thank you for that. So for that, check your notes to make sure. But for the book, it says pasteurization will be in our exception because it uses heat to kill off most bacteria, but not all, actually says. And sterilization is to get rid of all. But I think, yeah, autoclaving is definitely one. And then in your notes, you'll also see UV light and ionizing radiation. So the exception would be pasteurization. Yeah, because on first look, it's like, yeah, all of those get rid of some bacteria, but sterilization is getting rid of all of them. So yeah, pasteurization should be your exception. Okay. We'll keep that up. 14 says, in eukaryotic specifically, not prokaryotic, so kind of like humans or yeast or other eukaryotes, mRNA, in eukaryotic mRNA, which of the following are involved in RNA processing, so processing of the mRNA, I think they're specifically talking about, and it's choose all that are correct. So A says we have that 5' guanine cap. B says we have that poly 3 or 3' poly A tail or a polyadenine tail. C says exons are removed, and D says introns are spliced together. Oh no, sorry, my bad. This is not on silent. Embarrassing. Okay. Anyway, so we have 5' guanine cap, 3' poly A tail, exons being removed, and then introns spliced together. So, and this is true as all apply, so we have to know exactly what's going on. So the 5' usually does have that guanine cap, or sometimes I see it as guanosine because they're talking about, because guanine is just the base, but it's also the base, the sugar, and the phosphate. But I think, but poly A also does the same thing, so adenine is just the base, but the actual nucleotide is base, sugar, and phosphate. I don't know if she's trying to be tricky with that, because I didn't get to talk to her about her review, but these should be fine as answers. So we do have a 5' cap and a 3' poly A tail that's attached to our mRNA. So those are going to be true. But C says exons are removed, and then D says introns are spliced together. How I remember it is exons exit the nucleus, so the exons are what's going to be spliced together, and those are going to be translated into a polypeptide. So saying that exons are removed and then the introns are spliced together, it's kind of backwards. So you're removing your introns and you're splicing together exons, so the exons can then move out and exit that nucleus. So we're choosing all that are correct, and the correct ones would be A and B. Because introns are removed and then exons are spliced together. So these are kind of opposite. Exons exit the nucleus. If it was talking about prokaryotic mRNA or just RNA in general, this all would be not true because they don't have that 5' cap and the poly A tail, and they don't have any splicing together. They make the RNA, and then it goes and it gets transcribed or translated into polypeptides. In prokaryotes, actually, one RNA strand is used to translate many polypeptide chains, which is different than eukaryotic. Eukaryotic, you get one mRNA strand and you get one polypeptide chain. So prokaryotic organisms are going to actually get more proteins per RNA strand, or more polypeptides per RNA strand. That's like one of those differences that she may talk about. But for eukaryotic, A and B is true, but exons are spliced together, introns are removed. Then we have 15. 15 says, all of the following are features of DNA except. So this is another exception. She likes exceptions, apparently. So we got to watch ourselves when we pick these answers. We're looking for the exceptions, so something that's not right or false. So let's look through these answers. A says there are strands that run anti-parallel. B says nitrogenous bases pair in a complementary fashion. C says the length is connected with phosphodiester bonds. D says the strands are held together with hydrogen bonds. And E is all of the above are correct. So if we just think about what we know about DNA, so there are two strands going anti-parallel in a helix, or a double helix, because they're two strands. So strands that run anti-parallel, that is true. They're like, so one runs 5' to 3', the other one's 3' to 5', so they're anti-parallel. B says nitrogenous bases pair in a complementary fashion, and that is true. We have the complements of each other, so C bonds with G or interacts with G, if we're thinking of it in that case, or C hydrogen bonds with G, A hydrogen bonds with T. If we're talking about RNA, then U would hydrogen bonds with A. But this is DNA. So we do have strands that run anti-parallel. We do have complementary bases, nitrogenous bases, so they have nitrogen in them, which is true. C says the length is connected with phosphodiester bonds. So when it's talking about the length, it's talking about in one strand what's holding those bases together. Or not just the bases, the nucleotides together, because the nucleotide is the base, the sugar, and the phosphate all together. And where's that picture? So when we attach those nucleotides together, we're going to kind of attach a phosphate to an hydroxyl group, a COH, which is then going to be attached to another hydroxyl group in COH. So we're going to end up getting a C bonded to an O, bonded to a P double bond O, bonded to an O, bonded to a C. And that is going to be, so an ester would be C double bond O bonded to an oxygen, and then another C on either side. That would be ester. Diester would be the C double bond O with oxygens on either side, because ester on one side, ester on the other. But in this case, we have a phosphorus instead of a carbon with the double bond O. So we have P double bond O, 2 oxygens, and then 2 carbons on the other side. So this is a phosphodiester bond. So phosphodiester bonds do make up the backbone of DNA, or are present along the strand of the DNA. So it's important that they say the length is connected with them versus the 2 strands are connected. Because each nucleotide is attached by those phosphodiester bonds, but each of those strands are connected by base pairing with complementary bases, and specifically hydrogen bonds between complementary bases. So D says the strands are held together with hydrogen bonds, and that is true. So when you have G and C nitrogenous bases interacting together, forming a complement, they're complementary because G and C both can create 3 hydrogen bonds. And so they hydrogen bond with each other, they form 3 of them. A and T can only each create 2 hydrogen bonds, so they can also hydrogen bond together. That's kind of what helps make them complementary versus trying to match up A and C, or not A and, A and C or A and G or something like that, where they're not complementary and they don't go together. So this one's looking for an exception for DNA, but all of these are correct, so we don't really have an exception here. So our answer is going to just be A or E. All of the above are correct. Because in DNA, we have anti-parallel strands. We have bases that are complementary. The length is connected with our phosphodiester bonds for that phosphate sugar backbone, the strands are held together with hydrogen bonds between the base pairs. So all of these are correct when we're talking about DNA. 16 says, all the following enzymes are required for DNA replication, except. So A says DNA polymerase 3, B says helicase, C says primates, D says RNA polymerase, and E is topoisomerase. So all these are ACEs, all these are enzymes. So we kind of have to remember what each one of these does. DNA polymerase 3, we have 1, 2, and 3. 2, if I remember, elongates the leading strand. 3 is going to be a part of, I think, the lagging strands, and 1 is usually to help fill in those the fragments that you create on the lagging strand, to seal them together, to replace your primates. So DNA polymerase 3 is used for DNA replication because polymerase is usually like elongating DNA, and this is one of those enzymes that are going to help do that. C says helicase, so that's unwinding kind of, or breaking up your base pairs so you can get in and replicate those strands of DNA. So those are going to unzip the helicase or something like that, or unzip the helix is kind of what they say, but pull those strands apart. Primase makes primers. So these are RNA primers because DNA can't just attach to, the DNA polymerase can't just attach to DNA and start replicating. It needs that RNA primer in order to grab on and start replicating. So primase is creating that RNA primer so that the DNA polymerase can attach on and replicate that DNA. Helicase is going to, so helicase comes in, splits up the 2 strands. Primase comes in, creates a DNA primer, and then DNA polymerase is going to come in and elongate those strands after the primers are put down. RNA polymerase is used to elongate RNA, to make more and more RNA, which isn't really what we're doing in DNA replication. That would be more for transcription when we're trying to make a strand of RNA. That's when we use RNA polymerase. And then E, topoisomerase, I think this one helps with supercoiling because when you use helicase to pull things apart, we're going to have a problem with the twisting of the DNA since it's still in a helicoil fashion. So I think this assists with it, but it doesn't really, It's going to assist kind of like helicase assists with it. So the exception here that's not required for DNA replication would be RNA polymerase, because RNA polymerase is going to polymerize RNA or make polymers of RNA or make those RNA strands when we're doing transcription. But the other ones are required for DNA replication, so we can open them up and replicate them. Yeah. Do you have a question or? Okay, sorry, my bad. But do you have questions on this? Are you okay? So 16, the exception is RNA polymerase. That's used for transcription, not DNA replication. Okay. Then we have 17. All the following are true of translation, except. So translation is taking that mRNA, when we're working with eukaryotes at least. For prokaryotes, it's kind of just the RNA. They don't really have difference between, I don't think. Oh yeah, I guess messenger is coming from, not really the nucleus though, because prokaryotes don't have that. Anyway, sorry. Translation is going from your RNA to your polypeptide chain. So to go from RNA to your polypeptide chain, you need your mRNA with these strands. That's going to be read by the ribosome, and then you're going to have your tRNA coming in with those amino acids attached to them. So your mRNA is going to have the codon, or the 3 nucleotide segments. So on your mRNA, you have the codon, the 3 segment of your nucleotides. Your tRNA is going to come in, and the one with the matching anticodon, or the complement of your codon, is going to come in, interact, and bring in that amino acid. So they're going to use a ribosome to do this. So let's go through our answer choices. We're looking for an exception, so what's not right? A says all tRNA molecules enter at the A site. B says the polypeptide chain forms at the P site. C says tRNA molecules leave the E site. And D says translation ends when the mRNA hits a stop codon. So in our ribosome, we have the A, the P, and the E sites. So A site is where they attach. P is where the polypeptide chain is, where the elongation occurs. And E is the exit site. So A says all tRNA molecules are entering at the A site. That's mostly true. Most of them are going to come in at the A site, then move over to the P site and attach their amino acid. But it's not going to be true for the very first tRNA, for the very start of that polypeptide chain. So that very first one is actually going to enter at the P site. The next one, the second one, will enter at the A site, and then move over to the P site and attach its amino acid and make that polypeptide chain. So A would be our exception so far, that all tRNA molecules enter at the A site. So all of them do except for that very first one. The very first one's entering at the P site to create that polypeptide chain because it's the initial one. But let's say we didn't know that. Let's continue reading through these answer choices. B says the polypeptide chain forms at the P site. And that is true. So the P site is for the polypeptide chain where the amino acids are coming together and directed by those tRNA. C says tRNA molecules leave the E site, and that is true. E is for the exiting of them. So after their amino acid is attached to the polypeptide chain, the tRNA can leave and then go get another amino acid that matches their anticodon. And then D says translation ends when the mRNA hits a stop codon. And that is true. So after we have a stop codon, that amino acid is going to break off and no longer be attached at that P site. So we can't just continue adding more and more. So all of these are true except for A. It's not true that all tRNA molecules enter at the A site because the very first one enters that P site. Okay. 18 says, what kind of mutation results in no change of the polypeptide chain? So the polypeptide chain is going to be your sequence of amino acids. So this kind of mutation probably wouldn't be apparent from the functioning because you're going to still have the same polypeptide chain, you're still going to have the same protein, and it's going to be like nothing happened, except if you look at the genes of the person, then you can see the mutation there. It's not really expressed because maybe this type is usually where you have a mess up, but the mutation kind of codes for the same amino acid. That's kind of what this is describing. So it's describing a mutation where, although you messed up one of the nucleotides, you still get the same amino acid that it codes for. So nothing really happened. So it wasn't a big mess up. It was just a little mess up, and nothing kind of showed for it. Because of course, if you have a mess up and you insert a stop codon there instead, maybe it codes for a stop codon, then we're going to stop in the middle of our polypeptide chain and not create that full polypeptide. So there would be a change in that sense. Or if you messed up there was a mutation and it codes for a different amino acid, that would also change the polypeptide chain. So we're looking for something that's going to mutate our DNA but code for the same amino acid so our polypeptide chain stays the same. So our answer choices are A, a missense, B, a nonsense, C, a silent, and D, a frameshift. There's also E for none of these. I think think a nonsense might be made up. No, I don't remember if I've heard of this before. Missense kind of makes sense, possibly, if you think of what they are. But missense and nonsense don't really make sense because our polypeptide chain is the same, and it's not a messed up polypeptide chain. That's kind of what those would refer to. But I have a feeling one of those aren't even a real thing, and she probably made up a word, possibly. She likes to do that. Oh, it is? Missense and nonsense are both ones? No? Okay. Never mind then. Nonsense is the stop codon. Okay. So missense is when you have a different amino acid. Nonsense is the stop codon. Okay. I wasn't sure if those were the proper names for them. I need to review. But yeah, so you can kind of look at the word, though, and understand that that's probably not going to have no change in your polypeptide chain. So if your polypeptide chain has a missense mutation or a nonsense mutation, it doesn't really make sense that it would give you the same polypeptide. It would be an unchanged form, something that you wouldn't notice. D says a frameshift, so that's kind of where you're missing something, and you have to kind of, instead of having the right codons, you kind of shift over one nucleotide because you're missing one. That would probably mess up almost all of your nucleotide, all of your peptides, all of your amino acids with a frameshift. So that's also not a way where you're going to have no change of your polypeptide chain. But C, the silent mutation, is the one that makes the most sense, because this is going to be where you don't have a change in your polypeptide, so you don't really notice that there was a mutation after all, because it coded for the same amino acid. So this mutation, this isn't an exception. This is a straight one. So what kind of mutation results in no change in the polypeptide chain? It should be C, the silent mutation, or the one that's just not going to be apparent because it codes for the same amino acid. Okay. And then 19 says, which of the following is a possible genome of a virus? So A says single-stranded DNA, B says single-stranded RNA, C says double-stranded DNA, and D is double-stranded RNA. So one that you may be most familiar with would be that single-stranded RNA. That one's definitely one that maybe you're most familiar with for a genome of a virus. But actually, the genome of a virus could have single-stranded, it could have double-stranded, and it could also contain DNA. I know, I think the MCB virus is one of those examples that contain DNA, and they can also use that DNA to make RNA and that sort of thing, so they contain both of them. So in this one, which the following is possible, it should be all of them are possible for the genome. You also have those retroviruses, which take their RNA and create DNA from them, and then you create a double-stranded version of it. And viruses like the MCB virus, and like I think there's some more examples that she might talk about in her notes about ones that also involve DNA as well. So you can look at those as well, in case she wants you to know examples. Because I know her questions are usually a little bit tricky and sometimes very specific, so it's good to review maybe that portion if she goes over examples. So 19 should be E. 20 says, what type of virus uses reverse transcriptase? So reverse transcriptase sounds like it's an enzyme that's transcribing in reverse. So transcription is usually DNA to RNA. So if we have something that's reverse transcribing, it's going to do the opposite. It's going to go from RNA to DNA. So it goes backwards. It goes in reverse. So let's look at our answer choices. Our answer choices is A, Oolitic Virus, B, a lysogenic virus, C, a retrovirus, and D, none of the above. So A, Oolitic lytic virus, a lytic one, or the lytic cycle that is probably more familiar with, is where the virus is going to kind of go to its host and then insert its own DNA or its own information into the host's genome, so that when the host replicates, the genetic information of the virus will also replicate with it. Usually things that go through the lytic cycle can also jump over and go through the lysogenic cycle. So like a lysogenic virus or something that's going through the lysogenic pathway is going to be one that's going to lyse a cell. So it's going to produce more viruses and then break open the cell and kill it. So usually things that are lytic or viruses that go through the lytic pathway can then go over to the lysogenic pathway. They can just go straight through the lysogenic pathway and kill the host right away. But this doesn't really say anything about taking something that's RNA and creating DNA from it. But C says something that's a retrovirus. So retro kind of sounds like something that's retro, something that was way back when. So it's like kind of going reverse. This is kind of the type that will have that reverse transcriptase. It will have a retrovirus will usually have its own RNA, but also this reverse transcriptase, so it can create a DNA template and kind of insert itself into the person's genome. So lytic and lysogenic are just not specialized enough in terms for this, but retrovirus is definitely the one that has that reverse transcriptase in it. So when we talk about which one has reverse transcriptase, it'd be C, retrovirus. Then we have 21. It says, what kind of DNA transfer involves the use of a sex pilus? So we're looking for something that's going to directly attach to another bacteria and then send information through that. So a pilus is usually, they only have one pilus versus many fimbriae and things like that. So a pilus is usually the longest type of attachment that a bacteria usually has. So we have transformation, transduction, and conjugation as our answer choices. Transformation and transduction. So one of these involves absorbing kind of foreign DNA that's kind of floating around and just taking that in and incorporating that into its own genome. Another one involves a virus inserting its DNA into the bacteria. I'm not sure which is which, so if someone could look that up, that would be great. But one is absorbing floating around genetic information, so it doesn't have a vector to specifically give it that DNA. The other one is, I think transformation is the one with the virus, and transduction is the one where they just accept DNA, but I'm not sure. So maybe check that in your notes. But conjugation is going to be the one where usually it's the transfer of a plasmid from the bacteria from one to the other. And we're talking about plasmid, so usually you have something that's F positive and the other that's F negative, or plasmid positive, plasmid negative. And then you would transfer that by replicating the plasmid and then giving one of them to the other bacteria. So the pilus is usually used, so like from one bacteria to the other, direct transfer is through conjugation. I think transformation was when a virus comes in, transforms its genetic information and gives it some more DNA. And then transduction is where it takes in random genetic information. So for 21, it should be C. And not all of them. So not all of them are going to use that pilus. We ended a little bit early, but if we have questions either about these problems or about other information that could be on the test, you may other than that, I think this is all she gave for us to do today. I'm having a session that's tomorrow in Cape Florida. I think it's in Cape Florida. Let me check. But I'm a microbiology tutor, so that's why I took over this session today when Jessica was out. My review tomorrow is at 11:30 to 1:30. It's in Cape Florida. So hopefully I'll see you there. I'll have 30 questions that I made up based on her notes. So these are the 21 questions that she had for her review. Also, if you would remember to take the survey that we have here. They're at the ends of the table, so you can either use the QR code or the link to go to the survey and kind of share your thoughts on study union and the session you may have. Yeah? What? Oh, yeah, sorry. Yeah, sorry, not tomorrow. It's on Friday. My bad, sorry. Thank you about that. So my review is on Friday, the 29th. It's 11:30 to 1:30. It's also going to be live streamed and recorded, or just maybe probably like recorded and posted. This one was live streamed and recorded, so you can see this again on the YouTube Stark page. I'm also going to have my review posted on, not the SI Google site, mine will be on the Study Union Google site, the UCF Study Union one, which doesn't really come up in a Google search, so you can go to the Stark Facebook page and find that link if you need. But other questions, I saw other hands and then they went down, and then, yeah. But yeah, I'll take other questions if you have them as well. Yeah. The review on Friday is going to be in Cape Florida, which is the 3rd floor of the student union, in the bigger room. Okay. Yeah. From this review, or from the one on Friday? From this review, this one's on the SI Google site. So usually if you look up UCF SI, or even UCF SARC, you'll get to the SI one. The one that I will be posting for Friday will be on the UCF Study Union Google site, which is the other one. And that one you kind of need the link to get to it, which is kind of weird.